There we are. All right. So this lecture is going to give us a framework now of the cell itself. We're going to talk about the components of the cell. We're going to spend a lot of time focused on different organelles. But what I want you to do is I want you to keep in the back of your mind that the cell is a complex dynamic system of integrated components where the components are all working together simultaneously to give us life. All right. So even though we're focused on one particular thing at a time, understand that everything's happening at the same time in this very lively environment. Okay. All right. So what I'm going to do is I'm going to start with this video, and let me think. If I think I want to share the sound too. Let me see if this works. All right, maybe you guys could be able to hear this. And just sit back and enjoy. Can you hear the sound? Pretty profound, huh? The first time I saw that was in this video. It held up pretty good over time. I think I saw this in 2002 or 2003. I went to this conference up in Boston. I was working for a small company at the time, and we were doing some cool technology stuff. And so it's called SIGGRAPH. That's the name of the annual conference. SIGGRAPH is a conference where people go to present leading edge technologies that won't hit mainstream technology stuff for years. So in other words, back in 2002, I was playing with multi-touch screens that span the entire length of a whiteboard. I saw holograms and interactive tables, just really cool stuff. And at the end, at night, they have these movie nights where they show these these short features like what you just saw on screens like 5 times the size of a normal movie screen. And up to that point in my life as a molecular biologist, you just don't see molecules moving like this, right? And so this was kind of like a religious experience for me the first time it came on with that music too. And being able to visualize the cells inner workings like that, it just jumps you up a level, really. And then VR is taking it to the even next level right now. So imagine being able to be in VR in that environment, right? And then being able to understand things. So before this point, you had to read a book and then look at a cartoony picture of things that were all static. You had to like use your imagination for things. Now, there is a few important points to be made. First of all, the inside of a cell is not this big, spacious, aquatic, oceanic world. In reality, the inside of the cell is jam-packed. Everything is just packed together. Think of like a mosh pit and it's frenetic movement. Everything's just always jiggling. I'll show you a more accurate video in a little while. Oh, actually, I'll show it to you right now because it was at the end of the last. It was at the end of the last PowerPoint, and what I traditionally do is show what I just showed you first, and then I show you this one. So this is perfect. Let me just jump over to the membrane chapter again, and then I'm going to go all the way to the end here. Yeah, there it is. It's protein packing. And let's see this. So I have to share this with you now. All right, so it's the same people. It's the same people that did the first video, BioVisions here. But what you're going to see now, and I just want to warn you, this video gets a little creepy, simply because it unintentionally triggers some human psychology in the realm of eeriness. You'll see why, but it's definitely worth watching. All right, but yeah, definitely. Oh, I just realized, Emily, are you still there? You just have your camera off?
Sorry, I'm actually eating, so I just don't want to eat on camera.
Yeah. No, hey, that is awesome. No problem. As long as you're enjoying this. Okay, good.
Mm-hmm.
All right, yeah, you can, as you were. Sorry, you can just. Yeah. All right, here we go. Let me turn the volume up too. Okay. Make sure the resolution's maxed out here. Nope. Now it is. There we go. Oh, this is gonna be crazy. Can you guys hear anything? Maybe it doesn't have sound. So what you're seeing here is a vesicle that is covered in a protein called clathrin, which you'll learn about in cell biology someday. Right, but here's the thing. These are all proteins. Remember I told you everything's always jiggling at the molecular level? This is a more accurate vision here. What this is showing you is the physics that happens at this scale. This is how fast things actually move. This is, so you're looking at Brownian motion, and because of the way things are shaped, and because of where the electrostatic interactions occur, these things all happen spontaneously. So what you're seeing there, that's the actual motor protein you watched casually walking last time. It's burning ATP. You can see the ATP flying in here. There's that little thing that's fluttering around there. These little, here, let me pause it for a second because it's just so fast. This thing right here, that's ATP. That's ATP. This right here is the motor protein. The blue up here represents the phospholipids of a vesicle, okay? The yellow represents integral membrane proteins. And down here, you can watch the conformational change occur in the presence of ATP. You can see how the protein actually changes its shape. Right, and that's how you get that walk. It's all being driven by the energy of hydrolysis of ATP. The green, that's called clathrin. You don't need to know this for years still, but you see how it looks like it's got 3 legs. It looks like this. So what happens is you get this mesh of this protein, and that's what causes, you can see, so you're watching a vesicle form, and then now you can watch as the clathrin uncoats the vesicle. These are called clathrin coated vesicles. All right, so I think you're observing, oh, that's the fade to black there. All right, so it's a little creepy, and I'm really I'm kind of surprised that it didn't have sound in there. Go ahead.
Sorry. It looks like a bunch of cotton candy having a party rave and sugar rush.
Nice. I don't know why. Let me just make sure my sound works. And I hate these ads for YouTube Pro. Why isn't my sound working here? That's why. No, that's not why. Yep, that's why.
Around here. I don't know if we want to show from one part
this
of the
empty rose garden
another.
because the reporters just cleared out of here. But this rose garden was just set up for all of these reporters to ask questions of the president, who came up to the podium and talked about what he's doing with respect to China. He talked about terminating the U.S. relationship with the WHO, the World Health Organization. But you're right, Brianna. Typically at an event like this is a perfect opportunity for the president of the United States to comment on something of the magnitude of what we're seeing right now.
Zoom recording. Let's just get back to class. All right. Okay, you guys can see? Can you see class or PowerPoint? Okay. All right. So what I want you to do is, after you learn cell biology here, go back and watch that video again and see how many things you can pull out of that video that you now recognize. So I imagine that you probably didn't recognize a whole lot yet, but you will when you go through this. Okay. Oh, I have to go back and I have to share my PowerPoint again. There we go. Okay. All right, you guys can see the PowerPoint. All right, so cellular life. Everything on Earth is alive. I'm sorry, everything on Earth isn't alive. Everything that is alive on Earth is made of cells, right? We talk about cell theory. All right, and fundamentally, there's 2 body types: the prokaryotic body type and the eukaryotic body type, and that's it. There's no third, right? If you need to put a third thing here, that would be viruses, and they're not technically living. Okay. The word prokaryote and eukaryote, if you look at the words, you break them down. That karyo, the underlying Greek there means kernel. Okay, karyon. And that's attributed to the fact that when we first looked under the microscope and saw the nucleus, it looked kind of like a kernel of corn, little nugget. All right, the EU in front of a word, depending on who you're talking to, it means either well or good. But one way to think of it for this is these are either the cells with sometimes people think it means true. But eukaryote probably comes from the fact that the nucleus is well defined. And so because it's well defined, you can see it. It's the true version of it. So eukaryotic cell is a cell with a well-defined nucleus. Okay. The pro speaks to the fact that these are cells from an evolutionary perspective that came first. So the prokaryotic cells arose, and then afterwards we have the eukaryotic cells. Okay. And so all of life on Earth is made out of either prokaryotic cells or eukaryotic cells. Within the prokaryotes, there's 2 completely distinct domains of life: the bacteria and the archaeans, and that's spelled wrong. Just want to spell. Oh, apparently that stopped my share. Hold on, let me just put an R in there and then share it again. It's interesting. Sometimes that breaks the share and sometimes that doesn't break the share, and I have no idea. which one, or what's going to happen next. Okay, they're archaea. That's how you spell it, archaea. And they look similar at first blush when you look under the microscope at them, but the archaeans actually have a very distinct biochemistry and genetics that are so different from their bacteria cousins that we put them in their own domain of life. All right, and then the third domain of life is the eukaryotes, right? So I like to call the prokaryote and the eukaryote superdomains. And then within the superdomains, you have the domains, right? So if you wanted to rewrite the tree of life or clarify it, and I do, just getting my pen here. So you could think of it like this. There's the prokaryotic cells and the eukaryotic cells. And then within the prokaryotic cells, there's the bacteria and the archaeans. Whereas in the eukaryotic cells, there's just the eukaryotes, right? And then within each of these, there are multiple what are called phyla. Oh, I'm sorry, before that, I'm sorry. Before that, there's the kingdoms. Like this, kingdoms. And the kingdoms of the bacterial domain and the archaean domain are still being argued about. And the reason is when you have a bunch of bacterial cells in an area, they very easily share genetic information, it turns out. So that when you think you found a distinct species, you'll find a member of that species with DNA in it from another species. And so it's more like this web of genetics. It's very complicated. And so the prokaryotes make it kind of hard to clarify things beyond this realm. We could do species most of the time, especially in the different parts of the world. But there was this one survey they did about 10 years ago where they scooped up ocean water and they found millions of new bacteria. There's so many bacteria, and they could not easily distinguish the different species because as they started sequencing them, there was so much overlap. It's a mess. Eukaryotes are easier because it turns out when you're multicellular, it's a lot harder to share genetic information. Although it turns out, as we're learning more and more recently, it's still possible. All right, so what I want you to do as budding biologists is I want you to no longer think of the family tree of life in this very simple, linear species coming from species, okay? This happens. I'm not going to deny evolution, but it's important to realize that layered on top of that seemingly simple family tree is this thing called horizontal gene transfer. Okay, and that's the idea that one species can actually get the DNA from another species through mechanisms that are still poorly understood. Okay, so it's called horizontal gene transfer. And this is something that it's an area of active investigation. It might have to do with viruses, who knows? Okay, so anyway, within the eukaryotes, they're pretty well defined. We have the plant kingdom, plants. We have the animals, the metazoans, that's the formal name for animals, which you'll see in your later texts too, the metazoans, animals, the fungus, and then everything else. And it turns out there are many, you could call them kingdoms. They all have their own distinct single ancestors, which, remember, that's the goal of these family trees, monophylogeny, single common ancestors. So you have things like the amoebas. You have these other guys that the closest to us relative-wise are called the choanoflagellates. We have the dinoflagellates. There's the, there's so many. I'm trying to think. The names escape me right now, but there's so many of them. All right. So these are the kingdoms of life. What we're going to do today is we're going to focus on the animal, because that's what we are. And we're going to talk a little bit about plants. We're not talking about fungus at all, and we're not talking about the protease at all. That's why you see these things as gray right here. Okay, so what I want to do is give you the generic components of an animal cell, and we're going to also relate them to plant cells. But here's what I need you to understand. This is very important. What I'm about to present to you is a very cursory overview of a much more complicated world that I can't teach you because it's complicated and you have to build up to it. All right. I'm still learning about things. We're still discovering basic cell biology. And it's exciting. It's really exciting. The newer textbooks have new things in it. It's getting pushed, but there's only so much that you can give to a first-year biology student. All right. So keep that in the back of your mind. You're not learning everything here. There's things remaining to be seen later. Okay. All right. As a very brief historic perspective, the concept that life is made of cells came as early as 1665. That's when Robert Hooke in the British Royal Society published his book Micrographia, where it was the first instances of microscopy. All right. Now, he was looking at a piece of cork under the microscope, and he noticed that the cork had these kind of very irregular, oh, there it goes again, very regularly spaced compartments that to him were reminiscent of the living quarters of monks that were called cells. Now it's happening constantly. It's just, there's no rhyme or reason to this. I'm just trying to draw a square. Wow, I can't even draw a square now. There we go. So a monk cell, that's what he called it. And it turns out he wasn't actually even looking at a cell at that point. He was looking at the remains that were left behind by the cells, the cell walls. Okay, but he coined the phrase cell. We give that to Robert Hooke. 200 years later, and then some, we finally get to the 1840s, 1830s. These guys, Schleiden and separately Schwann, okay, so one was dealing with animal cells, one was dealing with plant cells. And I would say Schwann was animal, Schleiden was plant. They both independently confirmed that even though all these different plants looked completely differently and behaved completely differently, down in the microscopic level, they were all made of some similar fundamental component, the cell. Schwann discovered that independently within the same year, a year later, for animal tissue. Animal cells are a lot harder to see than plant cells because they're much more chaotic. They don't have the cell wall. They're less defined under the microscope. But they both independently discovered this. And so cell theory was born out of their work together, that all life on Earth, the smallest level of it is the cell. Okay. And we've already explained why cells are so small because we did the membrane chapter, right? So you understand that now. So this is where we really hit home with this idea of surface area to volume. All right. And so remember, it's all about the speed at which we can get to a surface, the speed at which we can get to a membrane, either in or out, right? And so the larger a cell is, the more rapid its volume will grow compared to its surface area, to the point where if you get too big, you get too slow, and you can't respond to your environment. You go extinct. So the smaller cells were the ones that won. Too small, and you can't hold your DNA and your proteins and all that. Okay, and so there's a happy medium there. Right, so the key is being able to get into the core there, to get into the more surface area for exchanging things. Right, another way to think of it is if I had this blue square on the left, right, imagine you eat some food and you swallowed it like whole. You didn't chew well enough. The orange dots represent the enzymes that will eat it, right? It's going to take a while to digest it from the outside. Whereas if you cut it in half, you now have access to this area where you didn't before, where you can also digest it. This is why we chew our food. The idea, it maximizes the surface area for attack, for digestion. This is why, this is the difference between having a solid ice cube and crushed ice, right? If you want ice cold water and you want it now, you use crushed ice because that's more surface contact with the water with crushed ice, okay? So from a mathematical perspective, you can see here that if we take a single 4 centimeter cube and we calculate the surface area, which is just the height times the width times the number of sides, right? There's 6 sides to a cube. You get 96 square centimeters. Okay, so it's surface area of 96. We'll multiply the height times the width times the depth, okay, or the height, width, and length, we get 64 centimeters. Okay, so the volume would be 64. So we can see the surface area is a bigger number than the volume, which is good, right? We want as much surface area as possible, okay? Now, if we take that exact same volume, so we keep that volume of 64, but now we dice it, okay, into smaller cubes, 8 2-centimeter cubes, that surface area shoots up to 192. And if we do 64 cubes, so if we cut it 3 times on each side, that number goes up to 384, right? So from 96 to 192 to 384, we're in the same volume of space, but look at how much that surface area increased. Okay, as an extreme example, our lungs, if you take a human pair of lungs, an adult average lung, pair of lungs, and you calculate the surface area of all those alveoli everywhere in the lungs, all those little micro branches, it comes out to an area that's equivalent to the same flat surface of a football field. So every time you breathe in, that gas exchange is happening over the surface of an entire football field's worth of membrane, which is pretty crazy. Okay, so you can see here, when you do the calculations, we're talking about these ratios now. So we're talking about the surface area compared to the volume, all right? And the key is the bigger that first number, the better, right? The larger the surface area is compared to the rest of the cell, the more rapid you're gonna be able to do things, okay? All right, now as a physical comparison, the eukaryotic cell is about 10 times larger than a prokaryotic cell. For this reason, a prokaryotic cell lacks internal membranes, as we'll see. And because of that, its body is constrained to the laws of diffusion, which happen over short distances. And so they necessarily have to be teeny tiny. So the eukaryotic cell, if you just took its surface area and its volume, you would see that it's terrible. It's a terrible ratio. But it's made up for the fact that inside of it, at any given point in here, there's a membrane somewhere. And so there's massive amounts of surface area within the cell. Okay, this concept you can think of as called being, I can't do it. Here, I'll write down words. Compartmentalization, compartments, different regions. And each compartment in here has a distinct chemistry, a distinct series of enzymes, right? So you can think of each compartment in here as having a different community of proteins. It'll also have its own distinct pH, okay, its own microenvironment. All right. All right, so the first thing we're going to do is we're going to cruise through the more simple version of the cell, the prokaryote. We're going to start out and we're going to dig in. All right. So again, these are just generic examples. And that should be here. I don't know. I have to correct that, but I'm not going to do that right now. All right. So starting on the outside, we have these things called the cellular extensions. I call them that. All right. So in other words, we have things that jut out, protrude. The first thing is called the sex pilus, right here. And what this is think of it as a hollow tube that sticks out so that we can take DNA in these small little rings, they're called plasmids, and we can exchange them with other cells. So in other words, 2 bacteria will do this together. They'll both form that sex pilus and then they can transfer DNA. Usually it's a DNA thing. This is what I was talking about earlier about the ocean sampling and how bacteria seem to be sharing their DNA. Bacteria are very promiscuous when it comes to this sort of thing. All right, they're sharing their little, and there's these little circles of DNA called plasmids, which we'll learn about more later in the semester. All right, little rings of DNA that contain a gene or two. That's the sex pilus. Okay, The second thing here is called the fimbriae. The fimbriae are these sticky hairs that allow the bacterial cell to adhere to surfaces, like your gums or your teeth, or your esophagus, your epithelium, whatever, your skin. All right, so they just increase surface adhesion. Then there's the flagellum, singular flagellum, plural flagella. And at the base, right here, at the base, if we zoomed in there, if this was the cell's membrane, there's a component to the flagella that, think of it like a cement core. If you ever think of putting a pole in the ground for a mailbox or a fence, you have to put that whole little cement bit in there first and then stick the wood into the bit, right? That's kind of how this thing is. This is called the basal body. The basal body is a very rigid, thick component, and then upon which we have a much more flexible structure coming out. That's the flagellum. There are also things called cilia. All right, cilia and flagella are structurally identical. Cilia are small, flagellum are big. Cells with flagellum tend to have one or maybe two. Cells with cilia have thousands. Okay? And the question being that's unspoken is, do bacteria have cilia? I can't think of an instance when they do. They tend to have flagella. I'm not a microbiologist, though. I'm sure that there's an exception to that somewhere. But my guess would be no. But I don't have a true answer for you. And I'm okay with that. All right. So that's the outside of the cell. Now we're going to go, let's see. Let's go extracellular here. So we're still outside the cell, but now we're outside the extension parts. The first thing is we have the membrane, right? The plasma membrane, just like any other cell. It's made out of phospholipids, and it's got proteins in it and glycoproteins. But then there's a cell wall, all right? Now the cell wall of a bacterium has a different building block than the cell wall of a plant cell. You might recall that plants have a cell wall made out of cellulose. Okay, all right, hold on one second, please. Recording, here we are. All right, so we got the plasma membrane. The cell wall of a bacterium is made out of something different, right? So plant cells are made from something we call cellulose, which we learned about back in the earlier chapter. Bacterial cell walls are made out of something called peptidoglycan. Okay, peptidoglycan is a modified protein sugar. Just leave it at that. All right, and a lot of bacteria have a double plasma membrane. This is something your book's not going to teach you, but you should know about it. So here's how it works. Here's one plasma membrane, okay, and then in between it and the second plasma membrane is the cell wall in between here. All right, I'm sorry, the glycocalyx. Okay, so there's a glycocalyx sandwich. Okay, and my Bluetooth just disabled, just to let you know. I'm not going to be able to use my hearing aid to listen to you anymore. So if you need to talk to me, you need to type. And I'm sorry about that, but I don't know why it didn't charge overnight, but it didn't. I hope it's not a battery related thing. But anyway, all right, so here, hold on one second. All right, here we go. Yeah, so here's a picture that shows you the double membrane. Okay, so you can see here, there's two kinds of glycocalyxes. There's the capsule, which is a hard shell, but it's impervious to a lot of environmental stress. And then there's something else that it could be called the slime layer. The slime layer is goopy and extra sticky. And so bacteria that have the slime layer can stick to your teeth really easily, for example, but they'll desiccate, they'll dry out pretty easily. Whereas the capsule is more hardy, but it's less sticky. Okay, and you can see here too, there's the outer plasma membrane, there's the peptidoglycan, the cell wall, and then another plasma membrane, an inner one. All right. Now within the cell, so let's talk about, now we're gonna talk about the intracellular stuff now. Excuse me. There's what's called the nucleoid. Okay, the nucleoid is the equivalent of the nucleus, where it's just this concentrated group called chromatin. We touched on, oh, I don't think I gave you that word yet. Chromatin is what we use to describe the stuff made out of DNA and protein. DNA is never found naked in the cell. It's always complex with some proteins or another. So collectively, we just call it chromatin. So you would say chromosomes are made out of chromatin. All right, and so there's this big complex in there. In addition, there are these little machines we'll talk about more at length called ribosomes. For now, you can understand them as sites of protein synthesis. This is where proteins are born, and the ribosome. All proteins, forever. There's no other way to make proteins. A mesosome. All right, now conceptually these are a little tricky. So here, let me draw this out for you. So here's the cell's membrane. Okay, this normal plasma membrane, and right here I'm going to do this. You see that? It's like taking a balloon and poking your finger into it. So it's an invagination of the cell's membrane. What this does is it increases the surface area, right? You see that? We've now increased our surface area without increasing our volume. That's good. So let's say that there was an essential machine that we needed on the surface of the membrane. We now have all this extra place to put it. Okay, and as we'll learn, for example, the mitochondria, the inner membrane of the mitochondria has tons of these things we call the cristae, as we'll see when we do the mitochondria later, right? So the inner membrane versus the outer membrane of the mitochondria, that's how we build the mitochondria. So the cristae is an extreme example of this thing we call the mesosome or mesosome. And then lastly, we have these things called inclusion bodies. Inclusion bodies are just dense aggregates, generally of protein and maybe some carbohydrates and some lipids that the cell will use to metabolize for later. All right. Okay. So now that you can see it in a slightly different way, so the cytoplasm of a cell is the interior compartment. The cytoplasm is in a prokaryotic cell. The cytoplasm is everything that's in here. It's just, so we talk about things being in the cytoplasm. Everything lives in the cytoplasm in a bacterial cell. All right, the nucleoid is within the cytoplasm. The ribosomes are within the cytoplasm. All right. Okay. Now, here's an amazing story. And I'll try to keep this very brief. Prokaryotic cells eat other prokaryotic cells. All right, so you guys remember endocytosis, right? So here's a cell and here's a cell. All right, this cell here, cell A, is going to eat cell B by endocytosing them. Right, and then eventually what happens is a vesicle buds off and the cell lives inside that vesicle, and then the way it will digest it. All right. Now, over evolutionary time, once cells unlocked the secrets of oxygen, all right, so we had photosynthesis put a whole bunch of oxygen into the air. Before that, cells did not have oxygen, didn't use oxygen, didn't like oxygen. They fermented. And when you look at glycolysis, which is a breakdown of sugar to make some energy, these early cells were very inefficient at getting the energy out of a glucose molecule. They could just make 2 ATPs, as we'll learn about later this week. Okay. As oxygen got pumped into the atmosphere, some cells, so all cells were stressed, some cells figured out a way to evolve to not die from the oxygen. Okay. And my hearing aid just died, by the way. So, this is going to be fun. Anyway, so, yeah, it's a little disorienting. Just give me a second. And I have no idea how loud I'm going to, if I start talking really loud, just wave. Okay, So some cells, hold on a second. I'm going to go put it on my charger. All right. So we were talking about endosymbiotic theory. That's the idea here. So one cell eats another cell. All right. So here's a cell that does not live in the presence of oxygen. All right. It does not efficiently make ATP using glucose. It does a little bit. Here's a cell that does. It's more highly evolved in the presence of oxygen, and it can make 30 ATP instead of 2. It's rich. So this cell eats this cell because that cell's rich in energy. So that makes sense, right? And it turns out slowly over time, this cell got tired of being eaten. And so it started getting internalized, but not digested. And so it stayed there. It turned out comfortable environment, nice and warm, nice and safe, and it still produced a whole bunch of energy, right? It still produced a bunch of ATP everywhere in the presence of oxygen. So the cell that ate it benefits from this production of energy, and the cell that got eaten benefits from having a warm environment, safe environment. And so it's mutually beneficial. That's symbiosis, right? So the idea that it was internalized, endo, and that it was beneficial, this is how we believe the mitochondria arose and the chloroplast. Now, what's the evidence for this? There's tons. And I won't go into the details, but just quickly, all mitochondria and all chloroplasts have their own genome. And that genome is a small circular piece of DNA, just like bacterial cells. That's 1. 2, they're about the same size. That's easy. 3, they have their own ribosomes. They make their own proteins, some proteins. Those ribosomes are the size and simplicity of prokaryotic ribosome, okay? So if you look at the ribosome of a bacterial cell and compare it to the size and complexity of a eukaryotic one, they're very different. That's a little dramatic. It's more like, maybe like that. All right, but the point is that mitochondria and chloroplasts have the more primitive versions of them. All right, so there's always this little interesting nuggets of information that lead us to believe that they used to be free living cells that got internalized. Now, because of this, you had a cell that didn't make a lot of energy suddenly have a ton of energy. And because of that, it could expand and become more complex. And that's what gave rise to today's eukaryotes, okay? So the way you can think of evolution working here is you start out with the prokaryotes. They give rise to the first eukaryotes that were the protists. The protists were around first. Those protists diversified. And some of them stayed, a lot of them stayed around to stay the same. They were single-celled, right? The key here is these are all single-celled organisms. Some of them eventually figure out multicellularity. That gives rise to initially something called the opisthocons, which I told you last time, that give you the animals and the fungus. And then some of them give rise to the plants. All the others are still here though. There's like 30 or over 30 of them. That's the story of evolution in a nutshell. And remember we had Luca, the last universal common ancestor. Okay? So this here, this jump from the simple to the complex, that's called set theory. Serial endosymbiotic theory. Serial because it can happen more than once. Okay, and now today there are some protists, some algae, where you had a eukaryotic cell. So here's a nucleus, here's a eukaryotic cell, eating another eukaryotic cell. He eats him so that you get this kind of weird thing. You get a nucleus with a nucleus. And then this thing shrivels up and becomes kind of a new weird organelle that we see in things like golden algae, which you'll learn about in some other class someday, maybe. All right, so you have you have the endosymbiotic evolution by which eukaryotes eating eukaryotes. Okay. So that's prokaryotes. Now we talked about the evolution of eukaryotes, and now here we are. And we'll talk about us today now. So we're going to talk about animals and plants. All right, so even though the cell itself has a low surface area compared to volume, it addresses this by this concept called compartmentalization. Okay. And animal cells are the only multicellular kingdom that has
It's from
you broke something.
I didn't.
Okay, I'll deal with that later, buddy.
I found it.
The base of a leg of some kind of piece of furniture. All right. Animals are the only multicellular organism that lacks the cell membrane, the cell wall. Okay. Basically, we traded protection for flexibility and agility. Animals are the ones that move, right? I mean, think about mushrooms. They have those mycelium networks. They move, but they're very slow, right? And really, they just grow. I think once they grow, they don't even move much. Plants can move slowly over hours, right? We can move here and the now. Right? So we don't have the cell wall slowing us down. All right, so we're now going to take our a survey of the animal cell. Here we go. So the first thing to think about from the broad brushstroke is there's two basic compartments. There's the nuclear compartment and the cytoplasmic compartment. Okay, most of the cell's work is done out in the cytoplasm. Whereas the nucleus is like Fort Knox for the DNA. Its job is to protect the DNA. That's one of its big jobs. All right. It's also a location where RNA is synthesized. And be careful here now. When we say protein factories, we're talking about the ribosomes. We're not talking about ribosomes making proteins. We're talking about making ribosomes. All right. Proteins are not synthesized in the nucleus. Hey, Kirsten. Yeah, so you want to avoid using laptops on laps and in beds and cozy things. You want to give well ventilated environment. Or it's the second you said it overheated, you froze. I was like, Okay, so the nucleus is the compartment where the DNA is housed, where RNA is synthesized, and where ribosomes are synthesized. Okay. Within the nucleus, and you'll see this dark spot right here, that's called the nucleolus. Oh my goodness. Nucleolus. Okay, think of it like the nucleus is nucleus. It's a tangled ball of chromatin, and that's where the ribosomes are manufactured. Okay? So RNA is only ever made in the nucleus, and proteins are only ever made in the cytoplasm. All right. So we're going to now go through a journey of the layers. Remember, all this is happening at the same time. So the first layer is membrane-bound components. All right, remember that we have all these different membranes. So let's go through the ones we're going to talk about. We have the mitochondria. We have what's called the ER, the endoplasmic reticulum, and we'll talk about the rough and the smooth. We have the nucleus. The nucleus has two membranes, like the mitochondria has two membranes. We have the Golgi apparatus, which is over here. And then we have various organelles throughout the cell with various names, things like the lysosome and the peroxisome. There are others that you won't learn about today. So these are the major ones. All right, superimposed on that, we have our chromatin, then the nucleus, right? So our genome. And you can think of the genome as having the chromosomes arranged in distinct territories called chromatin territories. And in between them are these little these little rivers. Okay? Canals, I like to call them, little nuclear canals. And at the end of the canals are these little portals that lead out into the world. These are the nuclear pores. And we'll talk more about those later. All right. Then layered on top of all that, we have the traffic of the cell, cellular trafficking. We have the vesicles. All right. So there's countless little lava lamp type blebby things moving everywhere. Layered on top of that, and really actually underneath it all, there's this scaffolding called the cytoskeleton. The microtubule network is the principal component of this. All right, and in the center right here, we're gonna call this thing the centrosome. The centrosome, as its name suggests, is a central body of the cell. As you learn this stuff, you're gonna see this root a lot, some. Some comes from the Greek word soma, which means body. Make sure you know that because you're going to see it everywhere. So the central body. The microtubules give the cell its overall shape. The microtubules are also the scaffolding upon which all the other organelles attach to. All right, so this gives us a substrate to live on. You also have a network of actin fibers, and generally the actin filaments, if this is the cell's membrane, they tend to live just underneath the cell surface. These are the cell stress response fibers. They're stress fibers. You can think of them like the muscles of the cell, maybe. They're involved in transducing signals across the membrane for communication. Okay? And then some cells, but not all cells, have another kind of filament called an intermediate filament, which, as their name suggests, is intermediate in size between the smaller actin filaments and the larger microtubules. Cells that are filled with intermediate filaments are strong. All right, so your armor, your skin and your nails and your hair is made out of what's called keratin. Okay, keratin is an example of an intermediate filament, very strong. Okay, your skin cells are all filled with that stuff. And then lastly, deep inside the nucleus, on the periphery of it, I should say, deep inside the cell, in the nucleus, but laminating the interior of the nucleus, is this thing called the nuclear lamina. The lamina is what gives an anchor point for the chromatin to organize around. And it also serves to give the nucleus its strength. Right, so there's a series of diseases called laminopathies, where people who are born with a mutation in one of the lamin genes have fragile nuclei, and it leads to all sorts of bad phenotypes. Okay, all right, and understand that cells don't actually look like this. This is what a cell would look like if it was flattened onto the surface of a petri dish. Right, in their native state, in your body. In any organism's body, cells take on a distinct three-dimensional shape, depending on what they do. This is a very artificial look, but it's useful for teaching. All right, and this slide here is just for your own, if you printed up my PowerPoints or you're looking at them without running the PowerPoint, this slide would look like this to start with. It's just chaos, right? It's useful for me to teach with, but when you print this up, it'll look like this. And so this breaks all that out for you. That's all this is. All right, so now we're gonna go through a tour of the organelles and understand that organelles are defined by their function. There are different compartments of the cell. Most of the organelles in your cell are membrane bound. They're surrounded by a membrane. Some of them aren't. The big notable one that does not have a membrane is the ribosome. Okay, so the definition of an organelle is a physically distinct structure in a cell that has a defined function. Okay. All right, so we're gonna look now at the different organelles, starting with the nucleus. So this is a slide that I would usually spend some time on and give you a tour, but I'm just gonna dig right in. I'm just going to jump right into the specific organelles. And then once I go through the organelles, I'll come back here and we'll put it all together. So the first one we're going to talk about is the nucleus. The nucleus itself has two membranes. there's an inner membrane and an outer membrane. All right, so as you draw this, I'll tell you what. I'm going to use I'm hitting escape right now. It's not having the functionality here. Hold on. And I just realized I had the recording paused. So we just, for those of you who are watching this in the future, we just finished a tour of the nucleus and the ribosomes. And I'm not going to go back right now and do that at this point. Sorry. But you guys are all here, so you heard it the first time. That's good. All right. So proteins. You've learned a lot about these so far. What we're going to learn now is where we find them. Okay. The first place we find them is everywhere in the cytoplasm. Now I'm going to give you a new word here. The cytoplasm itself has two components. You've already learned the cytoskeleton. The cytoplasm also includes the liquid fraction, the liquid component. We call that the cytosol. So there are cytosolic proteins. And there are cytoskeleton proteins, right? So there are proteins in the cytoplasm. 1. 2. I'll tell you what, for number 1, let's also add the nucleus, just to be complete here. Oh boy. So in other words, cells floating around inside this, proteins floating around inside the cell. The second place we find proteins, as you already know, are in the membrane. Let's make that plural, in the membranes of the cell. So there's proteins in the nucleus, there's nuclear membranes, inner outer, there's proteins in the ER, in the Golgi, yada, yada. So those are the integral proteins. Remember, integral membrane proteins. And then the third place, the third fractional component, the third location is secreted proteins, right? Like antibodies get secreted from B cells to go and do their job. And then I guess just for being complete again, we'll talk about the mitochondria and the chloroplast. Okay. All right. So who makes these proteins? The mitochondria, chloroplast, ribosomes are made by mitochondria and chloroplast ribosomes. That makes sense. The cytoplasmic and nuclear proteins are made by free ribosomes. And the ones embedded in membranes and the ones that are secreted are both made by the rough ER ribosomes. Okay, so the ribosomes bound to the rough ER make proteins, synthesize proteins that are destined for secretion or destined to be inside a membrane. Okay, all right, back to the PowerPoint. Okay, you guys see the PowerPoint? Okay. All right, the smooth ER. Now here's the thing with the smooth ER. It's more complicated than we'd like it to be in terms of teaching. It does a couple of things. And so what you need to do is acknowledge it's more complex than you think, but for now, I'm just going to associate it with some basic stuff. So the first thing is it's continuous with the rough ER, right? It keeps going and then it becomes smooth. So there's no ribosomes. This is the birthplace of lipid synthesis. That's the biggest thing you can think of it as doing. That's a primary function. We make lipids here. Okay. In muscle cells, there's a specialized version of the smooth ER called the sarcoplasmic reticulum. That's a repository for calcium. All right, so it's a predominant, so it plays different roles in different cell types. That's one way to think of it. Okay. In the liver, the smooth ER's job is to help break down toxins. So alcoholics, people who drink a lot of alcohol, the reason why they build up a tolerance to alcohol is because their liver starts producing more and more and more smooth ER to metabolize the ethanol. And so people that have a tolerance to alcohol have that tolerance because their cells have all this extra space to break it down quickly. Because it's a poison. Okay, so that's the smooth ER. That's the ER. Okay, now the Golgi. So now we're gonna finally break away from the nucleus and all the things attached to it. We're now out in the cytoplasm and we're gonna go across this little gap. We're gonna go to this thing called the Golgi. All right, so if this is an, hey, let's go back to the marker board now. I'll draw this for you. So if this is, let's go back to our cell that we've been drawing. Oops, go back up here. Now here, I'll tell you what. Let's do this. Oh, I can't use my mouse for that. Do this. Oops. I really do like this program, despite some of its flaws. There, let's give me some space to keep drawing this thing. All right. The Golgi apparatus is, so there's usually, so there's one. All right, and so you see I'm drawing it a little curved this way? It's like a flat pancake. So that individual thing is called a cisternae. I'm not sure if your book uses that word though. I don't want to confuse you. Hold on. No, I remember. I think your book uses the concept of a saccule. So you might see cisternae elsewhere, but I think your book calls them saccules. Okay, so there's a couple of them. And so I draw the first two kind of bending towards the nucleus. You see how they're kind of like this, like a cupped hand? And then the middle one is relatively straight. And then the next one is a little curved the other way. Like that, there. That's pretty good. And there's not always whatever I drew 5 of them. It depends. Now, it turns out in newer versions of the books and all that, there's another compartment right here. Kind of draw it both ways. That's a ER Golgi transition point. But we are gonna ignore that right now and just keep things simple. But if you look on the internet and you dig around watching videos, you might see another component in between the two of these. Okay, so that's the Golgi. And here's how it works. Vesicles will butt off the ER, smooth or rough, doesn't matter. All right, the vesicles butt off the ER, and just like a lava lamp, they're going to fuse with the Golgi. They're going to deliver their cargo to the Golgi. Some of the cargo, here if we zoomed in here, some of the cargo will be proteins embedded in the membranes, right? Remember, there's some membrane proteins. Some of them will be destined for excretion. Running out of colors here. Anyway, so if they're destined for excretion, they'll go into the Golgi like that. And then what happens is each one of these saccules will bud their own vesicles off and go to the next one, such that eventually they will blossom out the other end. Okay, so there's this constant traffic of proteins going from the ER through the Golgi saccules until they emerge from the outer face. And there's a name for the different faces. Okay, so the face that's closest to the nucleus, that's the cis face. Remember cis and trans? Same two words. Cis is close in this case. And then on the other side, this side of the Golgi is called the trans Golgi. I can't talk right now. Thank you. Okay, go.
Time for me to give it to you. What do I do?
Okay, you can go put it on the counter for now. Thank you. Okay, so we have the cis face and the trans face of the Golgi. Proteins mature through this journey. Think of this like a UPS sorting facility or some kind of combination of a sorting facility and a factory where things get processed, right? One of the things that happens in here, and so this is one way to associate form and function for you. One of the things that happens inside the Golgi is something called glycosylation, which we learned about last time. All right, remember that glycosylation is the process of adding sugars to proteins and lipids. All right, and here's how it works. So if this is the inside of the Golgi, let's keep it very simple. There's one saccule for the Golgi. Okay, and I'll tell you what. I'm going to zoom out a little here. I'm going to make this thicker. And here's the plasma membrane. Okay, so watch this. Inside the Golgi, there are enzymes, glycosylases. As their name suggests, they add sugars to things. Okay, and here, let's say, oh boy, that's too thick. Let's say right here, there's a phospholipid, one phospholipid. And that enzyme, let's make the sugars green. That enzyme is going to add a sugar right there. Okay? And let's say here's an integral membrane protein. And we're going to add a sugar to that too, right here. Okay? So it makes sense, right? We're inside the Golgi, the enzyme's in the Golgi, we're going to put the sugar right there. Right? So notice the other side of that protein, oops, now I'm too thick here. The other side of that protein here, and the other side of the lipid, I guess, the other side of the membrane, the other leaflet of the membrane, lack sugars, right? You see that, okay? So there's no sugars on the other side, only on the inside. Okay, now what we're gonna do is, I can't animate this, but go with me here. Let's say we bud a vesicle off here. Okay, so I'm gonna bud this off now. So here's a vesicle now that contains that protein, but here's what I'm gonna do to make it visually simpler for you. I'm gonna just rotate the vesicle so that the protein's here now. You see what I did? Okay, so I budded it off and then I rotated it. Are you with me still? Okay. So that red protein, that red squiggle, this red squiggle is the same as this squiggle. And all I've done was I've just budded this off and then rotated it. So because proteins can float, right? Remember, so the protein basically floated all the way over this way. Just pretend. And then the same with the lipid. That little phospholipid here, let's put him right there. All right, and so both of these things have their sugar on it. So the green, it represents a sugar, right? Okay, now we're gonna fuse that to the membrane. All right, so here, let's copy this. let's move over here. All right, so here, this is kind of neat. I can kind of animate it. You guys can see me moving this, right? Okay, so as I butt up against here, and those two membranes meld together and it fuses, it would start to look like this. Right? And the key is this protein and this little phospholipid that both have their sugars on them are now facing out, right? So now if I straightened all that out, I let the fusion happen completely, it would look like this. Right? And now we have a glycoprotein and a glycolipid facing outward on the cell surface. So stepping back, you can see that inside the Golgi, we have these enzymes that add the sugars. They glycosylate the protein, they glycosylate the lipid. Those glycoproteins and glycolipids then emerge on a vesicle. And when they fuse to the membrane, the interior of the vesicle is continuous with the extracellular environment. And so the sugars stay on the outside of the cell, okay? And so did that make sense? All right, good, okay. So that's the Golgi. That's what's happening over here in the Golgi. So collectively, if I now add to the cell right here, if I add that's the cell's plasma membrane. If I consider, oh, you know what? So I'm just gonna very quickly mention the next organelle for the sake of continuity here. I'll make him orange. So this organelle I'm about to talk about emerges from the Golgi, just like the vesicles, okay? So this is how this matures, this vesicle. It buds off the Golgi and it becomes its own thing. This is the lysosome. Okay, remember that lys means break, and there's some again, body. So this is a breaking body. Lysosomes are filled with enzymes that break things down. Remember hydrolysis. So we have something called a hydrolytic enzyme. So lysosomes are sacs filled with hydrolytic enzymes. Put little dots in them like that. Okay? And what they do is they merge with either an incoming vesicle that maybe contains some food. Remember phagocytosis? Remember a vacuole will come in with some food in it? That lysosome will fuse with that and unleash fury. It'll take out, it'll start shredding everything in there. That's one thing it does. Another thing they do is, let's say we had an old worn out mitochondria. The lysosome will fuse with an old cell component that's membrane bound and shred it to recycle it. Okay, so that's what the lysosomes do. So collectively, here's why I had to give it to you like that. Collectively, the lysosome and the Golgi and the ER, so all of this, let me use a different color here. I'll highlight. So all of this, okay, the cell's membrane, I broke that in there too. The cell membrane, the lysosome, the Golgi, and the ER collectively are called the endomembrane system. Okay? So the nucleus is not part of the endomembrane system. And there's a few, the ribosomes are not part of the endomembrane system. The chloroplast, not part of the endomembrane system. And we'll learn about something called the peroxisome as well. That's not part of the endomembrane system. Okay, so ER, Golgi, lysosome, cell membrane. All right, back to PowerPoint. So there it is, cis-phase, trans-phase, the shipping facility, glycosylation happens. Okay. All right, the next one, so I touched on this, the lysosome. We can think of them as a garbage disposal of the cell. All right, now here's a key point. Plants do not have lysosomes. One of the things I want you to learn as you study this stuff is compare and contrast the animal cell and the plant cell. This is one of the big differences. They don't have lysosomes. Plants lack lysosomes. Now think about it. They don't need to break things down and shred them because they have another job they've already learned. They go to their central vacuole. Okay, remember, plants dump all their garbage in the central vacuole to increase that osmotic pressure. So they don't need to break things down to recycle them. They have a better job. Okay? All right, your book has a horrible typo in it. You see where it says here, house phagocytes that dispose of bacteria and debris? And honestly, I don't know if it's your book or if there was an older book that I took this from, but let's just make sure we're clear here. If you see the sentence, a phagocyte is a type of cell that eats things. So it's a nonsensical statement to say that lysosomes contain phagocytes. That would be like saying your heart contains people. What? It's that crazy. Okay, a phagocyte's a kind of cell. So saying lysosomes house phagocytes, no, it's crazy. And I can't remember if I took that sentence out of your textbook or a different textbook, because I use a couple. So if you come across a statement that talks about phagocytes, it's a typo. It's a crazy typo. All right. Now, when a phagocyte eats a bacterium, right? So if this is a eukaryotic cell, and here it is eating a little bacterial cell. So we now have, I'm going to go back. I'm just going to go back. I've gotten pretty good at swapping back and forth here. All right. When we have, so let's do this as a, it's happening over here. When we have a cell, so here's a big cell that endocytoses a bacterium. Okay, so it brings it into the cell. We now call this thing a phagocytic vacuole. It contains a bacterial cell or whatever it brought, maybe food. Okay, let's use the same color. Here's Mr. Lysosome filled with all of his destructive enzymes. The lysosome will fuse with this thing so that you get, it unleashes its fury and those enzymes go to town on the bacterium and just shreds the whole thing, digests it. Okay, so cells that eat other cells are called phagocytes. You'd learn that in your anatomy physiology class. And phagocytes, this is how once they eat the cell, this is the fate of that cell, right? And so now you can imagine that happens, right? Let's say the incoming cell looked like this, though. It had all those mesosomes, right? And so let's eat that cell. Oh, who's that look like? That's your mitochondria. It's got two membranes, and the inner one has DNA in it, right? So you can imagine this is exactly how mitochondria eventually evolved. All right, back to PowerPoint. Okay. So yeah, I said all this. So there's something called autophagy. As its name suggests, autophagy is a cell eating itself, self-digestion. Imagine horribly your stomach bursting and all the digestive enzymes in your stomach eating away at your internal organs. That's essentially what happens when lysosomes burst. Okay, that'll kill the cell. And sometimes that's intentional. All right, so here's the summary of the endomembrane system, right? You can use this for your own studying, but so here's a secreted protein pathway, right? So here's something going out, and you can see here's a little food particle coming in and being, so here's your food vacuole. Here's the lysosome budding off of the Golgi, fusing with it to break it all down, okay? You can also see the smooth ER right here, and you can see how it's creating lipids, right? So lipids get manufactured in the smooth ER. Here's the rough ER making its proteins, okay? And you can see it fusing, and you can see these proteins getting modified and tweaked and cleaved and whatever until eventually they go out. All right, cool. Your book talks about antibodies. We talk about antibodies during our COVID-19 stuff. Antibodies are proteins that are specifically manufactured by our immune system that bind to and recognize and bind to foreign entities. And they do different things depending on the situation. If you want to learn more about that stuff, you want to research immunology. All right, you want to learn, if you specifically, you would do a search for adaptive immunity and B cells. Okay, it's really fascinating stuff that I wish I had time to teach it to you. I don't. Okay, this next organelle is weird. The peroxisome, I like to call the bomb squad of the cell. So here's the thing. Your mitochondria is making all these very toxic, high energy byproducts. Think of a dirty coal plant. We need the coal plant to make energy. We need our mitochondria to make ATP. But at what cost? There's all these toxic high energy products. And so the cell has to deal with these things. One of the ways our cells deal with them is by bringing them into the peroxisome and basically breaking them down from high energy molecules to low energy molecules. All right, so I'm going to give you an example of one of these things. If we were doing the enzyme lab, which we're not, but I hope that some of this is represented in the Labster stuff. So there's a high energy molecule called hydrogen peroxide, H2O2. You've heard of H2O. You have this probably at home in your little brown container that should be cold in the refrigerator because it's unstable and breaks down. Okay, so hydrogen peroxide that's hydrogen peroxide, H2O2. This is a very reactive oxygen species, or an ROS. Reactive oxygen species can tend to form free radicals. Okay? So if this thing bumps into another thing, it'll probably strip electrons from it. Okay, it's very reactive. Now, what happens is hydrogen peroxide, I should say, let me think for a second. 2 molecules of hydrogen peroxide, yeah, 2 molecules of hydrogen peroxide will break down into a water molecule plus molecular oxygen as a gas. Make sure it's all bound. No, 2 water molecules. There we go. Right, and make sure I balance my equation. There's 4 hydrogens, 2 oxygens, 3 oxygens. Darn it, I forgot how this is balanced now. I think it's 2. Oh yeah, no, wait, there's 4. Yeah, no, it's balanced. I'm second guessing myself. Okay, so here's the thing. This is a spontaneous reaction going this way, not fast, right? The peroxide that you have in your cupboard, if you go there right now and you picked it up, it would probably feel bulging. It would probably be bulging until you open the cap and it released a little. That's because you have a slow buildup of the oxygen gas in there from the natural breakdown of peroxide. Okay, so this is a very common molecule inside cells, hydrogen peroxide. It's a common byproduct of a lot of metabolic processes, and it's dangerous. All right, so like ethanol, I was telling you earlier that the smooth ER breaks down ethanol, right? So we break down ethanol or any other poison. All right, so let's say that we have all these bad things, all these poisons. All right, and technically they're not poisons, they're just dangerous molecules. Let's dangerous. Dangerous molecules. Sometimes they're intermediates from a metabolic reaction, whatever. What the cell does is it has all these different pathways to handle them. And what they do is through these pathways, they get down and funnel them all to hydrogen peroxide as one of the products. Okay. And then the very last thing we do to get rid of this is there's an enzyme called catalase. And the reason that's such a weird name is it was the very first enzyme ever discovered. Right? Remember, enzymes are protein catalysts, catalase. And what catalase does is it facilitates that reaction that I talked about earlier. Okay, it speeds it up. It does millions per second. So fast that you can actually observe the foaming action. Have you ever had a cut and poured peroxide on it and watched it foam? That's exactly what you're seeing there. That foaming, that's pure oxygen gas. Okay, and it's an old myth that if you pour peroxide on a wound and it bubbles, it means you're infected. That's not true. Your blood is filled with this catalase stuff. It's everywhere because it's a protective thing, right? The way I think the myth came from is the fact that when you get a fresh cut and you pour peroxide on it and it foams, what you're doing there is you're killing bacterial cells. That's why you pour it on there. This stuff is very dangerous, especially at high concentrations like what you have in your bottle. All right, so you pour it on your wound, you kill everything. So you clean your wound, you pour peroxide on it, you dry it, and then you put some antibiotic ointment on it and you put a Band-Aid on it, right? That's what you should do. And then after a day, you form a scab, right? So if you take the Band-Aid off and you pour peroxide on it, then you're not going to see any foam because you have a scab. You've sealed the blood into the body again. If you see foam, either you took it off too early and you don't have a scab yet, or your body's not able to form a closed scab because it's infected, right? That's the key. So now you are infected, right? So the foam there, the foam isn't because you're infected. The foam is because you haven't sealed your wound up, okay? So your body produces, you have specific cells. We have cells in our immune system where along the periphery of the cells are these little vacuoles filled with hydrogen peroxide. Okay, these are killer T cells. If this is a bacterial cell, that T cell will bind to it and unleash that hydrogen peroxide. It's chemical warfare and kill them. Okay? So inside our cells, we don't want peroxide everywhere, right? We want to make it go away. And so, going back, oh, I'm in the PowerPoint. Wow, that was working pretty well, wasn't it? And so, wait, you guys can see this, right? Can you see PowerPoint? Okay, so the peroxisome's job is to get rid of very high energy free radicals, okay? And a lot of it is peroxide, but there's all sorts of other bad guys, it's called. Okay, so they're called oxidases. An oxidase, so there's hydrolytic enzymes in lysosomes, there's oxidase enzymes in peroxisomes, okay? And their job is to detoxify. They also help break down long chain fatty acids, okay? Like triglycerides, I guess. And I like how and purines, remember purines are the nitrogenous bases in DNA and RNA. And remember, we don't eat those on purpose. This is why. They're dangerous. Purines are dangerous molecules that have to be gotten rid of because of all the nitrogen they contain. All right, so think of peroxisomes as the bomb squad or as the poison eater. Okay, now there's a whole class of things called vacuoles collectively. A vacuole generically is just a membrane-bound compartment in the cell that, mostly empty space, or it's simple, right? So when we bring that food particle in, that's a vacuole. When we bring the bacterium in, that phagocytic vacuole, all right? In plant cells, the largest, most obvious feature of a green plant cell is its central vacuole. You can see it over here, this huge, big open area. It maintains cell shape. I told you that's where it dumps its waste. And in plants that are poisonous, plants that have evolved poisons to counter herbivory when an animal's trying to eat it, it can't run away from the animal, right? So it's got to come up with a different way of preventing itself from getting eaten. Some of them have evolved poisons. Some have thorns, prickers, whatever. Some get poison. The poison lives inside the central vacuole. So the cell itself is protected from the poison, but when you eat it and you burst it all, now you get poisoned. Okay? So that's the central vacuole. There are other kinds of vacuoles. Okay, we learned about this yesterday when we talked about paramecium. Remember, he pumps the water out, the water expulsion vacuole, the WEV. And so you can see here, another simpler way to call this is a contractile vacuole. All right, so osmosis fills the vacuole with water, and then they're surrounded by actin filaments that act like little muscles, and they squirt it out. All right, you can see right here, you can see it looks like it's farting. I mean, it squirts it out, right? And then it gets thin again. And so that's how the cell prevents itself from exploding from osmosis. All right. All right, so that's vacuoles. All right, and then this is another plant heavy one called a plastid. Actually, these are unique to plants. All right, so a plastid is a storage organelle. All right, so and there's different kinds of plastids. So a leucoplast or an amyloplast would be those found in potatoes or corn. They store those starch granules we saw when we studied carbohydrates. Chromoplasts are housing the pigments of colors from flowers. All the beautiful colors and flowers, those ultimately can be traced down to these little plastids containing pigment molecules. It's pretty cool. So you now know the secrets of why plants have color. And then chloroplasts are specialized plastids that make sugar. Okay. All right. So there they are, chloroplasts. That was a good segue. So chloroplasts and mitochondria, I put these on the same slide because these are the two organelles involved in energy metabolism, the building and the breaking, all right? And each one of these really gets its own special chapter. Chapter 9 gives us the mitochondria and cell respiration, and chapter 10 gives us the chloroplasts and photosynthesis. All right, so for this chapter, for the cell biology chapter, just know their basic components and their function like any other organelle. Now, because they're complicated, they have more complicated structures. All right, so let's just go through this as a cursory view of it first. So chloroplasts, you can think of as sugar factories, little food factories. All right, this is why plants don't have to take food from their environment. They don't have to eat. So you need sunlight. They collect sunlight. They collect carbon dioxide. They collect water. Those three things, and they use those three things to generate sugars. And as a waste product, oxygen. Okay. Mitochondria, in the presence of oxygen, will bring in high energy molecules and extract their energy to make ATP. And I'll teach you all about that on Thursday. Okay, so you can connect it all with a single simple chemical formula. You got sunlight, you have carbon dioxide, you have water. Okay, and if for those chemists among you, you can balance this by putting a 6 in front of each of those. And that yields, we could say glucose, I can't, wow. There we go. Glucose, so C6H12O6, one molecule of that, all right, plus 6 molecules of oxygen, 6 molecules of carbon dioxide.
Hey, Dad.
And don't worry about the energy right now.
Do you know where Mom is?
You can't talk right now. Sorry. Okay. Okay, so and this is going to be the focal point of chapters 9 and 10. All right, so for now, gross anatomy of a chloroplast. You have big membrane on the outside, smooth membrane on the outside, and then another membrane on the inside that's also smooth. All right, so chloroplasts have two smooth membranes. And then inside that space, we call this space here the stroma, okay? And then within the stroma is this like city, this underwater city, I like to think of it as. You can see it down here. And so each individual housing unit is called a thylakoid. And they are made out of a membrane, and that membrane is continuous with itself. So you can think of all the different housing units have tunnels between them. So the thylakoid has a membrane, and it has an interior space. Okay, the thylakoid space, the thylakoid membrane, and this will be that will govern everything in chapter. The first half of chapter 10 will be all about that. They also contain their own DNA. Remember, that's housed somewhere here in the stroma. Okay, that's all you need to know about the chloroplast for now. And they're only found in plants. They're only found in plants. There are photosynthetic protists, but they're you can think of them as having a heavily modified version of this. I mean, really, the whole cell is almost a chloroplast. For bacteria, cyanobacteria that do photosynthesis, they are basically a big glorified chloroplast, and we'll see those when we do chapter 10 too. Okay, so that's the chloroplast. Nothing new here, really. Plant cells have multiple chloroplasts. I guess that's the take home here. You can see plants don't have one chloroplast; they have a couple, maybe 10 or so. All right, and this more photosynthesis. We'll deal with this when we do photosynthesis. That's enough for now. Okay, the mitochondria. So the mitochondria, like the chloroplast, has two membranes. The outer membrane, like the chloroplast, is smooth. The inner membrane of the mitochondria has a whole bunch of these very complicated folds called the cristae. All right, so outer membrane, and then the inner membrane. And how did that not generate any lines is beyond me. And then in the center here, the space in here is called the matrix. All right, so the matrix is the fluid compartment of the mitochondria. The analogy being the stroma right here. The stroma is the fluid compartment of the chloroplast. The matrix is the fluid compartment of the mitochondria. Okay? You can add ribosome here too. Actually, you can add ribosome to this list here too. So there's ribosomes in here. So there, you can see the little, oh, there they are. There's the ribosomes. And there's ribosomes in here. All right. And you can see there's this DNA. Okay. I'm looking at this slide right here, or this picture right here. So euglena is a protist. And here's the thing, protists are weird. You're gonna learn all these basics about cell biology, and then you're gonna go and study protists at some point, and you're gonna see that like there's all these exceptions to all these rules. So in this case, this picture right here, that cell has a network of mitochondria, right? So like they're connected somehow. I know nothing about that, right? But look, I mean, it's amazing. You can see a reticulated network of mitochondria themselves, right? Yeah, that's endlessly fascinating. Euglena also has a an eye spot. There's a little eye. They're photosynthetic, but they're free swimming. And supposedly, if you put euglena in the dark, it kills their chloroplasts and they become predators and go around eating things. Yeah. They're like, oh, look, imagine the first people looking at this under the microscope. Oh, it's green. It must be a plant. Wait, it's moving. Why is it eating that other cell? I don't know what this thing is, right? Nature doesn't care what we think. It just does its own thing. All right. And then here's a fun video of why the ATP, why there's, okay. So I should preface this with something. The cristae, all those finger things, they house a transmembrane machine. It's not a protein, it's a machine. There's like, I want to say 15 or 20, 30 proteins in there. And their job is to make ATP. And here's how it works. It just is a fun video. You see the rotating motor. Okay, this is ATP synthase, which we'll learn about when we do cell respiration later. These reside within the mitochondrial membrane, within the inner membrane, within the cristae. And so the more cristae you have, the more shelves you have to house the machine. That's one way to think of it, okay? Yeah, that's a motor. That's a mechanical motor being driven by protons. It's almost like a water mill. There's protons that flow, I can't draw on this. There's protons that flow downhill, basically, like water flowing down a river, driving a water mill, or wind blowing along a windmill. Protons drive that. It's pretty amazing stuff. All right, we're on the home stretch here. The cytoskeleton now. We've touched on this earlier. The cytoskeleton is the filaments within the cell. All right, they give the cell its shape. They give the cell its ability to respond to the outside world. They give it the ability to anchor itself into the extracellular matrix. And there's three basic kinds of fibers. Okay, big fibers, medium fibers, and small fibers. That's the way to think, organizing in your head. The big ones are called microfilaments, which might confuse you, the word micro meaning small, but here we are. Microfilaments are the big ones. Then there's intermediate filaments, which makes sense. They're in the middle. And then there are the microtubules. Those are the smallest. Okay? No, I'm sorry. I'm just reading from my slide. Hold on. You can, hold on. Let me fix that. Smallest to largest. And I'm gonna add actin filaments, known as microfilaments. That's why it's confusing. Yeah, so I'm using the proper terminology, but I'm just cleaning my slide up here. Send it back. Send you back. There we go. All right. New and improved slide. Here we go. Okay, so the three fibers. There's the actin filaments, which your book refers to as microfilaments. They are the smallest, okay? Then there's the intermediate filaments, and then there are the microtubules. Those are the largest, okay? So actin filaments, intermediate filaments, microtubules, and then the actin filaments are sometimes called microfilaments, okay? And so we're gonna go through each of these individually. So let's start with actin. So actin's the smallest one, about 8 nanometer diameter, and they're simple polymers of a single protein called actin. Okay, so like remember my little magnet ball? So here, this is what actin looks like, kind of. It's just a ball of amino acids bound up. It's a simple tertiary protein, no quaternary structure here. This is called globular actin, okay? You take this and you put it together with other ones of its kind, and you form these big, long filaments, and you do that twice, you have basically a double helix of actin. That's its tertiary structure. And that's what you're looking at over here. So you'll see two, just cross your fingers, And that's basically what an actin filament structure is like. Okay, so these are typically found on the surface, just underneath the surface of the plasma membrane. So if that's the plasma membrane, they're just underneath here. Okay, we call them the stress fibers. We'll also see them later when we do mitosis. It helps pinch the cells apart. Okay, so that's the actin. And here's an electron micrograph showing you the actin filaments. These also are responsible for amoebas moving. They're also involved in our muscles moving. Okay, so there's motor proteins involved in these. We'll talk about that at the end of here. All right, so they look kind of like muscles if you stain cells specific to actin. This is called immunofluorescence. It's a really fun and satisfying series of, it's fun work to do. I did a lot of immunofluorescence. You can take an antibody that's specific to any protein, conjugate it to a fluorescent marker, and then stain cells with it. And see all sorts of cool stuff. All right, so that's actin. Intermediate filaments. There's not one intermediate filament. This is a diverse family of proteins that all have the same basic structure. You can look over here. This is the basic structure of its tertiary structure. Okay, there's sticky ends, and then there's a coiled filamentous domain in the middle. And the way you do this is you take that, it looks like a barbell. And so here's a barbell. You take two of them, and you put them together, so you get a dimer, okay? And then you take that dimer and you put two of those together staggered and you get this tetramer, homotetramer, all right? And then this becomes the functional unit of building these protein filaments. And you can see they look kind of like, like potato sack, hemp. It looks like hemp rope or twine. Okay, they're really, really thick and knotty. And these provide strength to any cells that have them. Your hair is made out of this stuff. Your nails are made out of this stuff. Your skin is filled with this stuff. Okay, so the example is keratin. All right, so that's intermediate filaments. And here is immunofluorescence showing a typical fibroblast, which is a cell from your skin. Somebody took a series of pictures in 3 dimensions and then put them all together in an animated GIF. And so you can see 2 cells on the left there containing their intermediate filament network. And you can see that big black dark spot in the middle, that's the nucleus. So you can see that the intermediate filaments, the keratin probably, is not found in the nucleus, right? But you can also see that's gonna give the cell an insane amount of structural integrity, right? That's why you can pinch your skin and pull and contract it because it's just, these fibers are in there. Oh God, dry throat. Okay. So they're found in hair, they're found in feathers, scales, fish scales. Okay, all right. Microtubules, these are the biggest ones. The microtubules subunit is, so what do? They're found throughout the cell. They serve as the roadways upon which we travel, right? So we use those motor proteins to walk along these things. And all eukaryotic cells have something called the centrosome, which is construed as the microtubule organizing center, or the MTOC. Okay, it's almost always found right next to the nucleus. So if you're doodling, if you're doodling a cell, and here's the nucleus, you would draw the centrosome somewhere like right next to it. And then you could just radiate spokes out from there to get microtubules. They are made out of a dimer called tubulin. There's alpha tubulin and beta tubulin. 2 different proteins, so it's a heterodimer. And that, so this is the quaternary structure of the dimer. This is a quaternary structure of the microtubule itself. All right, so these are hollow rings. When I have these in class, I can make a microtubule out of my magnets simply by creating a very long, I'm not going to do it right now because it takes a while, but if I sat here and I created a very long chain of these, and then I just wrapped it around itself, I really can't see that. It's too short. You can begin to see the beginning. There it is, right? So you form these really long, beautiful cylinders. So that hollowness gives it both lightness and strength, okay? And the key to a microtubule is if this is the centrosome, microtubules build outwards. They build outwards from the centrosome, all right? Such that if you have the centrosome and a microtubule coming out of it, if you sever the connection, if you cut it here, the microtubule from this point inwards, this way, will depolymerize. It'll fall apart. Okay, so microtubules are not permanent structures. They're constantly being built and broken over and over again, as opposed to your bones, which are pretty static, right? Okay. Yeah, so you can see this, it's like a tangled ball of yarn in some cells, it looks like that. And also, as a point of note, in this cell, you can see in the nucleus here, see how it's blue? See the blue? That's DNA, that's chromatin. Notice that there's bright areas and dark areas. All right, the bright areas, because they're fluffy, they can take in the stain. That's your euchromatin. So the bright areas are the active parts, and the dark areas represent heterochromatin. They're tight and dense, and they can't take in the stain. But that beautiful picture right there, you can see that's very characteristic of a healthy eukaryotic cell nucleus. This is during mitosis, as we'll learn about in next week. These filaments here reorganize into a structure we call the spindle that grabs the chromosomes and yanks them apart. Okay, they basically orchestrate the logistics of mitosis. There's just another picture of it. Okay, so the inside of an animal centrosome. So here's the centrosome. The inside of the centrosome has these two very interesting structures called centrioles. All right, a centriole you can think of as a short cylinder made out of microtubules. And there's two of them, and they're always, we've been doing so good here, PowerPoint. They're always found at 90-degree angles to each other. And they're supposed to be the same size and shape. That's terrible. There's not a thick one and a thin one. Let me try that again. There. They're always found at 90-degree angles. Okay? Now, this is how you build, yeah, I'll tell you what, I'll use my whiteboard for this. This is one of those minutia that a lot of students get wrong. So let me try to give this to you in a good way here. Okay. So remember, we have, you guys can see my whiteboard? Okay. Oops. All right. So just recall, we have alpha and beta tubulin, and those will come together to form this kind of helical structure. Okay, so I'm gonna now cartoon that out to be something that looks kind of like a big straw. Okay, all right, so we're gonna take a cross section of this now, and we're gonna look down at it. So here, do you see that? We're looking down at the cross section. Okay, so that's what, when I draw a circle, that's what that is. We're gonna take 3 of them like this. 3 microtubules, and put them into a triplet. All right. And then we're going to take 9 of those. Let me see if I can do this. I'm not always good at drawing this. I'm going to try. Five, 6, 7, 8, 9. Nah, not bad. Okay, so this is 9 triplets, and there's none in the center. So we're going to call this the 9 + 0 configuration. All right, so there are other proteins that support this, but those are not microtubules. Okay, that makes sense? So if we look at it from the side view, we would see like that, okay, with a central core of some other protein. So that's a centriole. They're strong. They're stiff, okay? And there's always 2 of them. Oops. There's always 2 of them at 90-degree angles to each other in the centrosome. These serve, we don't fully understand what they do, to be honest. I have my own theories, but I'm not going to get into them right now. It has to do with gene expression. They're surrounded by a gelatinous goop of tubulin, right? The alpha and the beta tubulin. And that serves as a building block for extending out microtubules everywhere. Okay? All right. So let's go back to PowerPoint. All right, so there they are. You can see an electron micrograph. You can see the 9 + 0 there, right here. And you can see that they're 90 degrees to each other too. All right. Now we can talk about cilia and flagella and microvilli, which are a type of, collectively, these are the cell's extensions. All right, so a microvilli is if I have an epithelial cell, it's like I just have to it's got to get it out of its system a few times. And then I suddenly can draw, okay. There we go. Nope. There we are. I got a few in there. All right, those are microvilli. They are natural protrusions of the cytoskeleton that push outward like that. Okay, that's the microvilli. The cilia and flagella, in contrast, oh boy, it's like a speed thing. If I go too slow, no, it's random. There's no pattern to this. Okay, I can give up there. I'm just gonna show you pictures. Yeah, no, I have to draw this. Let me go to the whiteboard. Okay, so if this is the cell's membrane, there is a structure embedded in the membrane, and here's the secret. That structure, called the basal body, is equivalent to a centriole. It's got the 9 + 0 configuration, just like it is a centriole. It's just in a different place. It was discovered under a different period of time. It was called something different. It has a different function, but it's physically the same thing. All right. And then stacked on top of that is a whole nother set of microtubules that when we do a cross section of this thing, these are doublets now. And there's 9 of those, but there's also 2 in the middle. There's a doublet in the middle. This is called the 9 plus 2 I'm sorry, 9, oh my gosh. The 9 plus 1. No, it's 9 plus, I'm right. The 9 plus 2 configuration. All right, what the 9 plus 2 configuration does is it gives flexibility. Right? There are motor proteins. If we go around the I guess membrane, if we do motor proteins between these guys, those motor proteins allow the movement of those microtubules against each other. So here, oh, I guess I use this. So really, the motor proteins are also between the microtubules themselves, right? So imagine I put my, that's right, that's how I used to teach this. Hold on, let me get a pencil here. All right, watch this. So the motor protein, yeah, this is rubber, this will work. The motor protein here is between the 2 pens, and I'm gonna rotate them up and down like this. I can't do it well. Right, so here I have this pen between these 2 pens, and what I'm going to do is I'm going to roll these 2 pens. No, I can't do it with that. I need something smaller. Oh, here. The cap to chapstick. I'm going to put the cap to the chapstick, which represents the motor protein, in between these 2. If I could do this. And I'm going to roll this. It's hard to do this too. What I really need is a Lego tire, right? As I roll this, you can see, there we go. The microtubules are sliding along each other. Now notice the bottoms of them are up and down. If I anchored these 2 in place, okay, and I forced this to rotate between this or slide between it, they would actually end up bending like this. You see that? That's what causes the zigzagging, and that's what causes the locomotion. Okay, that's how the cilia flagella move. All right, back to the PowerPoint here. All right, so you can see here, there's a whole bunch of motors in there. Okay. And there's, you can see the 9 plus 2, I'm sorry, the 9 plus 2 versus the 9 plus 0. All right. And then the microvilli, these are just, they punch through the membrane and they increase the absorption for nutrients in your gut or wherever they're found. And the proteins that are helping punch through are the actin filaments. You can see that right here. Actin filaments. All right. And you can see here's the I always tell you how the actin filaments sit underneath the membrane. You can see them now pushing up, and that's how you get to the microvilli. All right. So your villi in your epithelial, in your gut, your alimentary canal, the villi are the tissue, and then when you zoom into the villi, they have microvilli. So there's so much surface area here for absorption. All right, we're on the home stretch, the last 2 things. There are these proteins called motor proteins that burn ATP to change shape. All right, so picture a bunch of rowers on a what are those called? The fast little skiffs they use for racing, like the Dragon race down in Philly, right? So you have the a bunch of rowers. Here's the rower in this case, and what happens is this head changes shape when the ATP burns. And so the whole thing, like a Viking longship, that's what I'm thinking of. So the whole thing gets moved at once. The whole actin filament here gets moved as these things all change shape at the same time. Okay, and that's how you get muscle contraction. So the protein is called myosin. So myosin and actin work together to contract your muscles. Okay, so in addition, myosin, and there's also kinesin and dynein. Where are those? Yeah, here. Kinesin and dynein are proteins involved in moving things throughout the cell, right? So those vesicles, even though they look like they might be diffusing through Brownian motion and randomness, are actually being directed by motor proteins along microtubules. All right, so kinesin, and these are families of proteins. They're the kinesins and the dyneins, and their job, particularly kinesin, is to move these things within the cell. Dynein, I believe, is the one within microtubules themselves for cilia and flagella. Okay, but collectively, they burn energy to move. Okay, and that's it. These extra slides, I don't think there's anything. You can read these on your own. Here's some fun symbiotic cells, or symbiotic slide. Yeah, okay, cool. So that is the cell. I believe that the homework is up already. I'm gonna stop recording. If the